Thank you for this opportunity to present some of our recent work, and specifically an approach we've developed for individualized profiling of germline antibody V genes. I'll start out to point out how much we've learned from isolating monoclonal antibodies, and that's true both in terms of antibodies from chronically HIV-infected individuals as well as from vaccinated individuals. Isolation of antibodies at the monoclonal level have really shown us how unusual HIV antibodies are. They're extremely diverted from germline, and that is of course a consequence of a very extended chronic infection. So they have high levels of SHM in general. You can also see in chronic infection that these antibodies are often highly expanded. Individual clonal families are very expanded. In addition, certain antibodies, and specifically those that target some broadly neutralizing antibody epitopes, have unusually long HCDR3 region, more so than the average antibodies you find in the repertoire. Another unusual property is that many of these antibodies are glycan reactive. And of course, we're well aware of the fact that broadly neutralizing antibodies take years to develop. So in contrast, when you look at vaccine-induced antibodies, there are major differences. These antibodies are extremely polyclonal. There are many individual clonal families that they're not as expanded. And this is, of course, a consequence of a much shorter exposure of the antigen to the immune system and an invariable antigen, which is what we most of the time immunize with. So these antibodies have lower level of SHM, yet you can see antibodies ranging from 5 to 15% SHM at the amino acid level. Glycan reactivity is very uncommon, not perhaps impossible to achieve, but uncommon. And so far, vaccination has not resulted in broadly neutralizing antibody activities. So one of the major aspects that has been studied is of course the target epitopes that these antibodies bind to. And this has mainly been done from chronically infected individuals, but more and more now from vaccination studies. Another level of detail can be achieved if we apply next generation sequencing. So this is really to get a more global view of the antibody response to infection or vaccination. And it's really the combination of the two that makes it very powerful. If you combine next generation sequencing with information from your monoclonal antibodies that you have functionally confirmed are specific, then you have a very powerful system. So one of the aspects that has been studied to quite an extent already is how broadly neutralizing antibodies lineages develop. So these are the so-called successful lineages. But what we are also very interested in is when the antibodies that we can induce by vaccination, what are their properties and what is limiting their development into perhaps more effective antibodies. There are many additional questions about the biology of the response that you can study if you combine monoclonal antibody isolation and NGS. And that include aspects such as where do these antibodies, what is the fate of them, where are they archived, which immune compartment, what is their persistence in relation to each other, is there competition between different clones, and what are their isotypes. So these are the questions that we're very actively investigating. But for the purpose of this talk, I want to point out that to do this work, we really need a very good solid understanding about the germline VDDA segments that are the building blocks of these antibodies. And this is really what this talk is about. So I'll just briefly give some background on antibody genetics. And of course, the basis for antibody diversity is that we encode many different segments that recombine during the development of a B cell. And this is really this combinatorial assembly of these building blocks that gives us this enormous diversity that we have in the naive B cell repertoire. This was elucidated and awarded in 1987 to Suzumi Tonegawa. And at the time, this was really a feat to reveal how this all worked. So in the human heavy chain locus, we know that we have approximately 50 V gene segments and a number of D segments and J segments. I'll focus primarily on V segments for the purpose of this talk. These V segments are divided into 7 families, VH1 through 7, and that's the same in other non-human primate species. So there is in addition many different levels of diversity that at this loci, and one is allelic diversity. So if we just look at the V gene segments, you can be homozygote or heterozygote for any of these V gene segments. And this is just to give an example. I mentioned that we have approximately 50 building blocks, but you can be heterozygote for example, 20 of those and homozygote for 30, which would give you 70 alleles. So the number of alleles you have will vary, and it can be many more than the actual building blocks. That's one level of diversity. The other level of diversity is that through evolution, we have lost and gained some of these segments. So there are clear duplications and deletions at the genomic level of these V gene segments. So this, of course, adds complexity and makes the diversity in the population that we are now starting to understand. This is not very well represented yet in the current V gene databases, and the most common one is IMDT. So although this has been an incredibly useful resource, it is insufficient, and that is becoming more and more clear. IMDT currently, both for human V genes and those of other species, contain many non-functional alleles and a certain number of sequences that represent sequence errors. And this is probably a consequence of the fact that many of these sequences were submitted many decades ago, before sequencing methods were as robust, and they have not been independently validated. Also, they come from a limited number of subjects, so it's not at all a representation of the global diversity. So this is something that is really needed to look at in more detail. And apart from human and mouse, these databases are really very incomplete. So many of the model systems that we use for vaccine studies, there's very limited information in these databases currently. And this is important because when we study antibody evolution and we study responses to infection or vaccination, we need to know what we start from. What is the germline configuration of these antibodies? And for that, we need information about these germline segments. So how do we improve the databases? One aspect that has really sort of hampered this is that genomic sequencing is extremely time-consuming and low throughput. This is because the loci is large, very large. The heavy lambda kappa chain loci are large, and they contain so many repetitive sequences that assembling these is not something that you can do with normal whole genome sequencing approaches. You simply do not get sufficient coverage over these regions to be able to assemble the loci in an adequate way. So you need extraordinary sequencing depth, and this has really not been achieved. If you look at efforts such as 1000 Genome Project, there is it's not sufficient coverage over this region. And it became more and more clear to us that really we need an alternative approach, because it will take simply too long to sequence sufficient numbers of individuals to get a good representation of global diversity over these loci. So we started thinking more and more about how to apply next generation sequencing to achieve this goal. And what we can do here is that we can sequence the recombined repertoire. And this gives us then millions of sequences that we can use if we have an appropriate computational approach to sort of parse these data and identify germline alleles from these data. And we do this either by making the libraries through 5' rays. This is an unbiased approach that will capture any V gene segment. But often we start by that, and from that we can really study these upstream regions or in the leader sequences of each V gene segment. That is the basis for designing primers that we can then use in multiplex. And that's what we eventually then use for our library preparations. We're starting from approximately 1 million sequences that we run on the MiSeq, Illumina's MiSeq instrument, using the 2x300 base pair read approach. And I'm just mentioning this because we often get questions about 5' rays versus 5' multiplex. And the reason we do it in 2 steps is because 5' rays gives us this advantage of being non-biased, but the sequencing reads are a little bit longer, so you get a little bit less overlap. Fewer sequences where you get sufficient overlap to do the merged reads. And you can see here that close to 550 base pairs on average for 5' rays, whereas for 5' multiplex, it's about 500. And for 500, the overlap is not a problem. So with the multiplex approach, we then get sufficient library sizes that we can really have a good starting point for mining and defining germline sequences. The approach is rather takes a little while to explain, so this is just a very brief summary of how it works, and it's published. But starting from the IgM library, we basically assign all the sequences to a starting database. And there is then, through bioinformatic windowed and linkage clustering approaches, we can parse these sequences into different clusters, and this then identifies candidate germline genes. There's a number of criteria that is in this algorithm for this program, and they are described in detail in the paper. But in brief, we look for sequences, consensus sequences from these clusters that we then search for to find exact 100% identical sequences in the real database. And those have to be recombined with a certain number of unique CDR3s to pass our germline filter. So several hundred identical sequences that are recombined with many unique CDR3s. That's indicative of a germline sequence. If it's not recombined with many different HCDR3s, it could be a clonally expanded, mutated IgM memory. So once we find these candidate sequences and they pass the germline filter, we go through this several iterations to replace the original database. And once you find no more candidate sequences, you have basically identified all the germline sequences that are expressed at reasonably frequency. If they're expressed at lower frequencies, you have to go through more iterations, but it's exactly the same approach otherwise. And to just illustrate how this looks, starting with a small database of just one sequence from each of the VH families, this is how it looks as these individualized databases build up. Whether this is a human or an experimental animal, it's exactly the same approach. So starting with one iteration, you identify germline genes, you build up the library, the database from this particular individual until you find no more. And this is then the germline alleles that this subject has. And we've shown that there is significant allelic diversity between individuals, and this is why it would be very hard to do this by whole genome sequences, and why you need a higher throughput rapid approach. To some of the validations we've done, and this is just to show you how it can look if you sequence a small family as an example to look for the different allelic contents in these individuals. And if you take a father, a mother, and a child, you can see in this Venn diagram that the child has no alleles that are not coming from either the father or the mother. You can see that the child is homozygote for 37 alleles and heterozygotes for 30. And this just shows how different these three individuals, even from the same family, are. If you look at this is heavy chain, if you look at the light chains, there's a little bit less diversity and there's slightly fewer V gene segments for lambda and kappa, but the same sort of overall result in that you can very clearly see which alleles were inherited from which parent. We have done various steps of validations before this paper was published. Some include that we made libraries from different immune compartments from the same individual, for example, PBMCs and bone marrow, or from different time points from the same individual. And IgDiscover always generates the same end result. So whatever you have, you will have whichever compartment you take the cells from or which time point you look. So these are your germline genes that you're born with. Other ways to validate alleles would be to say that you find them in independent individuals, the exact same 100% identical sequence, and we do that for some alleles. And we've also done targeted genomic sequences, and this is perhaps the most important part because then you can be absolutely sure that they're present at the genomic level. This paper was published, as I mentioned, at the end of last year, and we've made this software available. It's an open source software that you can find at this link. That's where the source code is. A lot of information about the analysis criteria, as well as frequently asked questions. And we're keeping updating this site to make it available to the broader public or to researchers that are involved in these types of activities. And we appreciate any feedback from people that are starting to use it. One of the things that we, of course, are quite interested in is to really improve our knowledge about human V gene diversity. And one paper that points to this need was published by Catherine Scheepers in Lynn Morris' lab, where they identified many new alleles in a cohort of South Africans. This was published in 2015. And we're currently collaborating with the Morris Group to apply ID Discover to this set of samples, and both to validate their previous study and to also look for lambda and kappa diversity in these populations. So this is ongoing. We also have done a very small sort of preliminary survey of VH diversity just to see what we find if we take 5 individuals from different parts of the world, of different ethnic origins. And as you can see here, it's an extremely small study, but just to give us a sort of an initial flavor of the diversity. You can see in this Venn diagram that there is significant diversity. And of all of the alleles, about 23 are shared between these 5 individuals. And perhaps an even more interesting take-home from this very small study is that the number of unique alleles was much greater in the African individual than in the other individuals. And this really suggests to us that this is where we need to increase the numbers by 100-fold. We want to sequence and get good statistics on this. But clearly, we need to have a better understanding of alleles in groups that have not previously been well represented in IMDT, for example. We looked also specifically at VH1-2 alleles because there is considerable interest in these alleles because of course, because of VRC01 class of antibodies. And in IMDT, there are 5 alleles currently of VH1-2, and those are shown here at the top of this tree. We have never seen 203 and 201 expressed in any of the human libraries we've sequenced, and they have not been reported in the literature either. So these are questionable alleles and probably errors in the database. On the other hand, we have found 2 alleles that are not reported previously, and they were found in the African individuals that we've sequenced. And if you align these and look at the VRC01 motif, the 3 amino acid motif that's required for VRC01 binding to ON, then you can see that only 2 of the alleles have that motif. It's present in VH102 and 04, but it's not present in the other 3 alleles. So this is important, and it's the 05 finding has already been described by Leo Stamatados and Marie Pansera, but the other 2 alleles have not previously been described. We increased the numbers a little bit. This is really early and ongoing work, but if you look at 17 individuals now representing, again, different parts of the world, we just wanted to know what the frequency of having this allele is. And as you can see, 202 is the most frequent allele. But if you look specifically among those 6 samples from Africa we've analyzed so far, there is, it's not uncommon that they have these other alleles. And so we believe that this is important to define further and much greater numbers of individuals, not the least for ongoing germline targeting approaches that are attempting to elicit VRC01 class antibodies. So sort of in general, this is very early stage, but we feel like in the era of personalized medicine, where there is a focus to really understand biology and disease at an individual level, this is another part that needs to be filled in. There is a gap in our knowledge about alleles, antibody alleles, in diverse populations. And we think this is important for future association studies, not only for infection and vaccination, but perhaps also to understand autoimmunity or allergy, where antibodies also play a role. So far, the method that we've published, ID Discover, is designed to identify and find new alleles. And for that, our throughput currently, if we just use one sequencing machine, is approximately 12 individuals per run. This is, of course, relatively low throughput, although much, much higher than whole genome sequencing. And this is ongoing with the Caprisa samples and other samples that we have obtained through collaboration. But to really meet the need, we have also developed a high throughput approach. And this is just to assign sequences to known alleles. So here you don't need to have as many sequences as for the discovery. So here you can basically increase this by tenfold. So now this makes it possible to really sequence thousands of individuals. So this is really amenable to scaling up. And so with that, I really want to make the point that it is now feasible to generate individualized V gene databases from from really large cohorts of people, including vaccine trials, such as those that are ongoing, for example, in South Africa right now. ID-Discover is also extremely useful for generating de novo databases where there is very little information available. And this includes various experimental animal models where there is either very little information or no information at all. And one of the tasks that we've just completed is to generate databases for the guinea pig, and this is presented at this meeting. This all sort of ties in very well with efforts that are also ongoing in the field. And I'm referring to the adaptive immune receptor repertoire community that was established by Felix Breeden and Jamie Scott. And this is a working group that is actually convening regularly. And really the goal is to review and revise the way we identify germline alleles with next generation sequencing. We're sort of working in a different landscape than 10 years ago. And two of the major goals of this community is to identify alternative ways to define germline genes and how they should be validated, as well as define common standards for publishing antibody repertoire sequencing data in journals and elsewhere. And Martin Corcoran, who's a senior scientist in my group, is very actively taking part in this and leading one of the working groups on germline gene discovery. So to summarize, the approach that was developed in our lab, ID-Discover, does three main things. One is to identify entirely novel alleles, and the other is to quite rapidly now construct individualized V-gene databases, and to also make databases from diverse species where very little information was available before. So having this available is extremely important for drawing the right conclusions about antibody affinity maturation and sort of doing the biology that we're all involved in, namely to understand how B-cell responses develop from the naive repertoire to the mature antibody repertoire that hopefully will recognize and bind our antigens in an appropriate and effective way. And also just to say again that we've developed a high throughput approach, so we believe that this is now something that could and should be done in large vaccine trials so that you have the best chance of analyzing the data subsequently in the most appropriate way in terms of antibody affinity maturation, SHM levels, etc. And persons that have been really instrumental in this work are underlined here: Martin Corcoran, Ganesh Fad, and Nestor Vasquez-Bernard from my lab, together with computational scientist Marcel Martin. And this is all work that has been a spin-off effect of our HIVRAD, that's led by Rich White at the Scripps. And we apply it to our immunization studies, which I have not included in this presentation here. But it's all funded by the HIVRAD, and we have started collaborations with Lynn Morris Lab in Johannesburg, and several other groups is about to begin. So thank you.